Hey everyone, welcome to our lab iPlantMicro, situated at ETQB institution in the beautiful city of Oeiras, Portugal. We are a dedicated team in the realm of microbiology with a particular focus on studying plant growth promoting bacteria. These remarkable bacteria have been a game changer in agriculture, aiding our crops in dealing with various stresses and significantly enhancing their productivity. The role they play in sustainable agriculture is priceless, following the necessity for precise identification, and the most reliable method for this is through the barcoding of the 16S marker, which requires the extraction of high-quality DNA. Today we are thrilled to give you an insight into how we perform this sophisticated yet fascinating task right here in our lab. As you might know, bacteria are prokaryotes, meaning they lack a nucleus, which makes DNA extraction relatively straightforward. Let's walk through the process. So, we begin by adding 100 microliters of autoclaved milky or DD water into a 1 milliliter empatherf tubes. Why water? It's pure sterile environment that prevents contamination and aids in the initial suspension of bacterial cells. Then, using disposable loops, we transfer a small amount of bacteria into the water. This loop ensures that only we get the bacteria that we need without introducing foreign particles or other microorganisms. Mixing is essential. It physically disrupts the bacterial cell walls. This step is a form of mechanical lysis, using a physical force to start breaking down the tough outer layers of the bacterial cells. After, we do a quick 10 seconds vortex providing intense mixings, further aiding in breaking down cell walls. This vigorous shaking ensures that the cells are adequately disrupted. The next step is heating up at 95 degrees for 12 minutes. It's a critical step because it denaturates the proteins and breaks down the cell's membranes and the wall, fully releasing the DNA into the solution. The high temperature ensures that all cellular compounds are sufficiently broken down. This step separates the unwanted cellular debris from our target DNA. Centrifugation at 16K RPM causes the heavier particles like proteins and cell fragments to form a pellet at the bottom. The DNA, being lighter, remains in the supernet above the pellet. Then we have to recover this DNA. We then carefully transfer the supernatant, which contains our precious DNA, to a new tube. This step ensures we're collecting only the DNA, free from contaminants and other cell components. After completing the extraction process, we move on to assess the DNA's concentration and quality. We assure absorbance at specific wavelengths, 260 nanometers for DNA, 280 for proteins, and 213 for phenols. The ideal ratio from 260 to 280 absorbance ratio should be between 1.8 to 2, indicating high purity DNA. However, in cases where the DNA doesn't meet our standards, we don't hesitate to use commercial extraction kits. These are particularly useful for extracting DNA for more challenging bacteria. This ensures that regardless of the sample, we achieve the high quality DNA necessary for our research. We hope you found this technique for ECHOC DNA extraction interesting. If you have any questions, are curious about our work, or are interested in joining our lab, please don't hesitate to contact us. Here's our Instagram page where you can stay updated with our latest research and lab activities. Thank you for watching, and we hope to hear from you soon.